Lift up my computer. Really good. Can you lift up? Okay. Yeah. Might be better. Yeah.
Okay, Prof. Morisco, we will start the guest lecture. And MC will start this meeting.
Honorable Dean, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro, Honorable Head Department of Agriculture, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro, Honorable Head Agrotechnology Study Program, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro, Honorable Professor Mauriscu, PhD, as speakers, dear students and participants. Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Shalom, salam sejahtera bagi kita semua. Om Swastiastu, Namo Buddhaya, salam kebajikan. Ladies and gentlemen, welcome to guest lecture Sustainable Agriculture in Facing Climate Change Production of Civil Rice, organized by Agro-Ecotechnology Study Program, Department of Agriculture, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro. Singing Indonesia National Anthem, Indonesia Raya. Ladies and gentlemen, please stand up. Ladies and gentlemen, please sit down. Speech and opening by Head Agro-Ecotechnology Study Program, Department of Agriculture, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro, to Engineer Korno MAPPL SC BSc, time is yours.
Okay, thank you. Dear Honorable Dean of Faculty of Animal and Agricultural Sciences, Honorable Head of Department of Agriculture, lecturer, student, guests as distinguished guests, and also Honorable Professor Mauriscu from National Chiayi University, Taiwan. Good morning, all of you. Assalamualaikum warahmatullahi wabarakatuh. Dear all, I want to welcome to Prof. Maurice, the guest lecture of today. And before that, I want to inform that we are from study program of Agroeco Technology, Department of Agriculture, Faculty of Animal and Agricultural Sciences, Diponegoro University. We situated in Semarang, Central Java, Indonesia. For the program of agroecotechnology, we regularly invite guest lecture from both Indonesia and overseas. Last month in October, we already invited for guest lecturer from Indonesia. And this month in November, we invite two guest lecturer from overseas, and this is from National Chiayi University. Okay, last week we have Prof. Chuang, and today we have a special guest lecturer, Prof. Mauriscu. Dear all, we want to inform you that between Diponegoro University, especially our faculty, and Taiwan already built a collaboration program. In the past time before the COVID-19 pandemic, we sent several students and lecturers to Taiwan to do summer program, and interns. We also invite several students and lecturer from Taiwan to come to our department, our faculty, to do the same thing. This is a joint summer program. So we already have a collaboration between Diponegoro University and several universities in Taiwan. And today we invited a special guest lecturer, Prof. Mauriscu, to share his expertise in building C4 rice to cope with the climate change. Hope that this will improve our knowledge. All of us, our students and lecturer, and also guests that join this guest lecture today. We hope that by collaboration we will expand the collaboration to research in the future. And also one of our young lecturer, Achi Rosida, also graduated from Chiayi University, Taiwan. And also one of our alumni, Widi Dwi Noviandi, also takes master program in National Chiayi University. We hope that in the future, more and more students from our university to study in Taiwan. So, that's the dean already joined in this class. Maybe good morning, Pak Bambang. Good morning, Mr. Dean, Pak Bambang. Okay, I think that's my Halo, Pak Bambang. Could you hear me, Mr. Dean? Okay, I think we continue. I think that's my small talk to welcome Prof. Maurisku from National GIE University for guest lecture today. I think that's all. I give back to MC, please.
Okay, thank you to Professor Engineer Karno MAPPA SCPSC as Head Agroecotechnology Study Program, Department of Agriculture, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro. Speech and opening by Dean Faculty of Animal and Agricultural Sciences, Universitas Diponegoro to Professor Dr. Bambang Walio Hadi Eko Prasetyono. Time is yours.
Okay, thank you. Good morning, ladies and gentlemen.
Good morning.
Assalamualaikum warahmatullahi wabarakatuh. With all due respect, the guest lecturer, Professor Maurits SB to PhD. Good morning.
Good morning.
Professor Maurits SBQ, PhD from Bio Agriculture Department, National GIE University, Taiwan, and School of Biological Sciences, Washington State University, Pullman. And then the head of Agriculture Department, the head of Agroecotechnology Study Program, the moderator, Professor Kusmiati, the colleagues, the student, and all of the participants. But first, let's say thank you to God for His grace that we can gather in this occasion. The pandemic situation presents challenges and opportunities for the higher education sector. This makes the international academic activities requiring new program formulation. Online guest lecture or online visiting professor become new formulation in the current situation. This is an opportunity that can be obtained from the global pandemic situation, that the world is increasingly borderless. Students can very easily attend lecture from professor in another country, such as from National GIE University, Taiwan. Professor Maurichiku and participant, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro, is currently in a better ranking position in animal and agricultural sciences subject according to the version of Ministry of Education, Research and Technology, QS, and Time Higher Education world-class university rank. This condition is triggered by international mobility activities carried by the professor, lecturer, and students in terms of education, research, and community service activities. One of the contributing activities is guest lecture. On the other side, this activity has the main goal to provide students with broader knowledge and insight on a particular agriculture topics. In addition, by the presence of professors from foreign universities, further collaboration can be initiated both in education and research, so that in the future, there will be further program between of animal and agriculture sciences, Diponegoro University, and Bioagriculture Department of National CII University, Taiwan. When the COVID-19 condition has recovered, we are very happy welcoming Professor Maurits Pu to visit Diponegoro University located in the Semarang City, Central Java, Indonesia. Hopefully, one day it can be realized and then we can do further cooperation. And for all students and participants, please gain as much knowledge and new insight. It's a pleasure to welcome you all. With the name of God, Allah Subhanahu wa Ta'ala, officially, I open the program. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Professor Dr. Bambang Waluyo Hadi Eko Prasetyono, SD Faculty of Animal and Agricultural Sciences, Universitas Diponegoro. Next agenda is photo session. For all participants, please turn on your camera.
Okay, what should I start?
One, two, three. Thank you. The main agenda is the lecture from Professor Maurits Pu, PhD. This session will be led by the moderator, Professor Dr. Florentina Kusmiati. To Professor Dr. Florentina Kusmiati, time is yours.
Okay, thank you for the MC and the Honorable Dean of Animal and Agricultural Sciences, Head of Agriculture Department, Head of Agro-Ecotechnology Study Program, from Prof. Maurits Q, PhD, distinguished guests, lecturer, and student. Good morning. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera buat kita semua. First of all, I would like to welcome all of the audience at this guest lecture who organized by Agro-Ecotechnology Study Program. In this occasion, we would like to have a guest lecture from Professor Maurits Q, PhD. The lecture session will be divided into three parts. The first lecture is about introduction of the modern technology in agricultural research for crop improvement and the use of maize CLK transcription factor to improve rice chloroplast development, photosynthetic, and productivity. It will take around 60 minutes. And then after the first lecture, we will have a break for 30 minutes. And then we continue with the second lecture. It will discuss about productivity of C4 rice. And then after that, we will have a discussion session. Before the presentation begin, allow me to introduce the guest lecturer, curriculum vitae of Prof. Maurice. Prof. Maurice is a special distinguished professor at Department of Bioagricultural Science, National KII University. He is also an emeritus professor from School of Biological Sciences, Washington State University, Pullman. Next. And then he was graduate in 68 in agronomy from National Chongqing University, Taiwan, and he was continue his master degree at University of Guelph, Canada, and then he graduated in plant physiology at University of Wisconsin Madison, United States. Next. Oh, the professional experience is as a vice president at National KII University, Taiwan. And before that, he was a dean at College of Agriculture, National KII University. And he was also as a director and distinguished professor at Department of Bioagriculture Science at the same university. Next. This is the Professional Society of Professor Maurice Gu at the Scientific Research Society at American Society of Plant Biology and also American Association for the Advancement of Science. Also in who in biology of higher education in USA. Next. As editorial, Prof. Maurice also as editorial board in global chain of biology and then plant production science and botanical studies. Next. And his research interest in mechanism and evolution of C4 photosynthesis and engineering of C4 rice, plant tolerant to abiotic resistance, and then plant transformation technology. Next. And then at he had funded research from 2013 to 2022 in a rice improvement project from Sinica Academia thematic project. Oh, it's a lot of funded. Okay, next. And then he has a lot of publication in the journal. The latest one is in 2021 is in genomic with the title chromosomal level genome assembly semi-dualized Taichung native one in an initiator of Green Revolution. Next. Yes, more than 20 manuscript. And then in the last, he has also have a manuscript submitted. Next. And then he also prepare the new manuscript. We hope that this manuscript will publish soon. Okay, next. And then this is a patent application, but in Taiwan alphabetical, so it's difficult for me to read. Okay. Without further ado, I would like to welcome the guest lecturer to deliver the presentation. Prof. Maurice, time is yours.
Thank you so much. for the nice introduction. Yeah, in the last 16 years since I returned from the US and started my teaching at National Jai University, I have a lot interaction with Indonesian, not only the university, but also the industrial. Yeah, so it's a great pleasure for me to share my research with your university, especially we have outstanding alumni, Ochi, returning to your university. And right now, Widi is also with us. Widi, you want to say hello? Hello. Okay, yeah. So really great pleasure. And hopefully, as the Dean say, in the future, maybe we can collaborate not only in teaching or in research. I really enjoy collaboration with international scientists in the past. Australia, China, Hong Kong, also Indonesian, also Australia. So I really, even in South America, Chile, Argentina, I have some collaboration. So I really not only enjoy the scientific interaction, but also the great pleasure to visit different culture and people. Yeah, and my visit of Indonesia is also very, very enjoyable. Yeah, so I look at for future collaboration. Yeah, today I just want to share with you some of the exciting results from my laboratory. Okay, so let me just start it from my first lecture. So what I would do is in the first lecture, I'm introducing the modern technology used for plant research. And then in the second part, I want to share with you this very exciting result we just published in Plant Physiology. That is use of a maize transcription factor to boost rice chloroplast development, photosynthesis, and grain yield. And then in the third part, or the second half of my presentation, will be on generation of C4 rice. Okay, so let me just start the first one. Okay, full screen. Okay, should I minimize this one or just a smaller? Maybe just minimize. Yeah. Okay. All right. In this first short presentation, basically, I just want to introduce the modern agricultural biotechnology in crop improvement to the student and the audience, because I learned that the majority of the audience today is undergraduate student, starting from freshman, junior, and then senior. Okay, so I believe students in agriculture biotechnology must have the knowledge in these following disciplines. For example, the basic courses, students should be prepared to have competence in mathematics, chemistry, physics, organic chemistry, ecology, evolution, and even computer science. Most of our agriculture students do not get exposure to ecology and evolution. And I think that that's a mistake from my point of view, because we have to know how the ecosystem get established and how evolution process play the key role in modification of speciation. Okay, and then these days we are talking about automatic learning. So computer science is also very, very important for our students to face the future professional development. Okay, and then for agriculture, especially in crop science, I think the advances courses such as physiology, biochemistry, molecular biology, even soil microbiology, genetics, breeding, tissue culture, genetic engineering, and then the bioinformatics. These are all the key player in our research in the future. So I think if you are in the audience as an undergraduate student, you have to think your interest, what you like to be specialized in, okay? And then specialize in these discipline, and then using the knowledge to develop your research and career, okay? So this is what I think these are very important courses for undergraduate student or even graduate student to learn, okay? So we are facing a very drastic or dramatic climate change these days. So I think in the future, our student in agriculture, biotechnology, the major goal, I'm sure, would be sustainable agriculture. In other words, how to maintain crop productivity under climate change is very essential, okay? And we all know that there are two types of climate change. Abiotic stress, sometimes it's too hot, sometimes it's too cold, sometimes it's too dry, sometimes it's too much water, flooding, and low light could be some problem in some region. And then too much light that cause photo inhibition, or in the case, the soil may have heavy metal that reduce productivity, or even increased UV light due to ozone depletion. I happen to be close to South Pole many years ago, and they have a big ozone hole above their head. So they have a tremendous UV light that cause damage not only to human being, animal, but also to plant. Okay, and in some region, air pollution is a major problem that reduce crop productivity as well. On the biotic side, during climate change, the insect pathogen or soil microbe can be affected. So these are the all important area for agriculture student to deal with in the future. Okay, yeah. Okay, so what are the modern technology that we can use in plant research, especially to identify the gene that has an important function? Okay, now in early days, we go outside or go to the paddy field or some area to find a mutant, and then we bring them back to study what are the genes get impaired, cause the change in phenotype, okay? But we know that natural mutation caused by UV light or some other reason is very rare, usually 1 out of 1 million, so very low. And if you consider rice genome, rice have more than 50,000 genes, okay? So if the mutation rate is so low, we cannot identify too many mutants by natural cause. So then the chemical or physical mutagenesis become very important. We can use large scale production of a mutant using chemical or physical, like a radiation or mutagen, chemical mutagen. So you generate a big population of mutant that allows you to identify key gene for particular trait, okay? Bigger gland, smaller plant, bigger plant, bigger panicle, such such and such and so on. Okay, and then more than 20 some years ago, people started to use a transgenic approach to make a mutation, make it very saturated. For example, in rice, there are a few transgenic mutant library. There are so many mutants you can use to identify gene function. Sometimes it could be tagging by activation or inactivation. So in this way, you know the importance of a specific gene for a particular trait. Okay, yeah. And then with the genomic era coming up later, these days we are using transcriptome or proteomic study to profile gene expression at the transcriptional level or at the protein level. Okay, and then with advancing technology in DNA sequencing, genomic analysis or genome sequencing becomes so powerful that we can profile the gene structure, composition, and then their function. Okay, so these are the powerful tools for future agriculture scientists to use. And then for breeder, I think most of you are probably familiar with QTL, quantitative trait loci. Basically, you can link gene function and the trait, phenotypic trait. And by this genetic segregation study, you can estimate what are the most important genes responsible for the trait. Okay, and if a multiple trait, you can estimate which gene is more important than other gene in control the trait. Okay, so this is frequently used by breeder or genetist, the QTL. And then probably in the last 10 years, some powerful tool emerged, so-called GWAS, genome-wide association. Basically, for example, in rice, if you have a collection of 100 different varieties from different parts of the world, then you can do genome sequencing for all of them. And then you compare the difference between these different varieties in their genome structure, composition, gene structure, gene sequence. And so you can say in the rice coming from tropical area, which is a more temperature high temperature adapt, you would know which gene may be linked to this climate adaptation, or to a cold region, to a salt drought tolerance. Okay, so this is a very powerful approach these days. Okay, and then of course we all know that hybrid crop technology is so important these days. For example, hybrid rice developed in China and in some other country, because they have a hybrid vigor, so they tend to increase crop yield by 15 to 20%. In the seed industry, vegetable or agronomic crops, they always try to generate hybrid vigor, hybrid rice, hybrid maize, so on and so forth. So the technology in this area should be further developed. And usually, if you have variety from very distant genetic background, they tend to give you higher hybrid vigor, higher productivity enhancement. Okay, and then these days we are talking about marker assist breeding. In this case, in the screening in a in the screening of elite cultivar, if the trait has a known marker associated with this particular trait, then in the screening of the F2, F3, or progenies, you can just find the marker, and then that is linked to the trait. This make it very efficient. Okay, you don't need to grow the crop to know how they would perform. Basically, at the young seedling stage, you can tell whether this is gonna be a productive variety or not. Okay, and then also from the genome sequencing project, from many different variety, you can search for SNP. This means a single nucleotide polymorphism. So you can compare the genome sequence for a particular gene, how their sequence variation, and how that link to a particular agronomic trait, drought tolerance, cold tolerance, heavy metal tolerance, because if this gene is very important, and then its sequence get mutated only by single nucleotide, and it can cause impairment of the function or its function. So you have a different phenotype. Okay, so this is also very powerful approach in breeding program. And then lastly, I think that our student also should be trained to be competent in tissue culture for propagation or for genetic transformation. So genetic transformation can be used for gene function study. You can do gain of function by overexpression, or you can do loss of function by inactivation of the gene, and then see how they affect the traits. Okay. In the case of crop improvement these days, we know genome editing, especially using CRISPR technology, is very powerful. Yesterday, Japan announced that for the first time, they would release commercialization of tomato that was added by CRISPR for enhanced nutritional quality. Okay, so this is a big advancement, and many European countries consider that CRISPR added transgenic plant are not really considered GMO that require very extensive approval. Okay, so this CRISPR approach is really powerful. In our lab, we use CRISPR to alter the starch composition of rice, and we recently submit the paper to International Journal of Molecular Science. Okay, so hopefully in the future, I can share this information with you guys too. Okay, so for this part, I think that I want to stop here. Okay, and then I want to move on to my second part. Okay, so how do we proceed? Stop share. Stop. Oh, stop share. Okay. 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 Share screen again. So I want to move to the second presentation. Okay. Enlargement. So everybody is with me, right? Okay. Yes. Okay. Last week we got our paper accepted by Plant Physiology. The title of this paper was Maize GLK Transcription Factor to Improve Rice Chloroplast Development, Photosynthesis, and Grain Yield. And I think this is really exciting study, because this is the same approach can be used to improve many other crops for increased productivity through higher photosynthetic capacity. Okay, so let me go to the background. Yeah, so why we need to study photosynthesis and chloroplast in relation to crop productivity? Because it's very simple. Because photosynthesis account for up to 90% of the dry matter accumulate in plant. Okay, and then chloroplasts are the site for photosynthesis, and even more importantly, production of phytohormones, for example, ABA, and production of antioxidant. So chloroplast is not only for photosynthesis, it's also for stress tolerance too. Okay. Now, in terms of a gene related to chloroplast development, we know that there are two what we call golden two-like factors. These are transcription factor. These transcription factor act as a transcriptional activator for chloroplast development in both C3 like a rice and C4 plant like a maize. Okay, and then we know that rice, GLK1, GLK2 are also logs to maize GLK1 or we call G1 or GLK2 or G2 respectively. However, rice, GLK1 and GLK2 act redundantly to regulate chloroplast development in mesophyll cell. In other words, if you knock out rice GLK1, it doesn't impair chloroplast development, or you impair GLK2, it doesn't affect chloroplast development in rice leaf. You have to knock out both of them to get albino leaf. However, maize GLK and GLK2, GLK1, GLK2 are functionally specialized and expressed in different cell-specific manner. So GLK1 is specifically expressed in mesophyll cell, and GLK2 specifically expressed in bundle sheet cell. Therefore, the maize GLK gene may have acquired new function during evolution and confer maize a higher photosynthetic efficiency. And I think this mechanism occurs in most of the C4 plant. And because of this specialization of GLK, maize GLK or other C4 plant GLK, allow them to have high photosynthetic efficiency. So if we can use these gene evolve with a new function and express in C3 plant, we can expect to increase C3 photosynthesis, and productivity. Okay. So the research goal of the study is that in order to boost rice chloroplast development and photosynthesis, we generate 5 different transgenic rice plants that overexpress maize GLK1, maize GLK2 individually or simultaneously with a constitutive promoter like a ubiquitin or S35 promoter, or using maize's own promoter. Okay, and this maize own promoter for GLK1 gene, for GLK2 gene, and also for both of them together. So we have 5 different transgenic rice line developed with individual gene, with a different promoter or their own promoter. Okay, why we are doing this? Because normally gene expression has a cell specificity, tissue specificity, or development specificity. So if you don't follow these rules in the natural condition, then you may have some side effect. And if you look at the literature, expression of foreign genes in most transgenic plants were driven by constitutive promoter, such as maize ubiquitin or mosaic virus, certified as mosaic virus promoters. Okay, these are strong promoters. Yeah. So if you use these promoter, express your foreign gene in all tissues, then it may cause some negative effect on growth. Okay, so this is a major reason for our research design. Okay, so basically we want to compare expression of maize GLK gene in rice individually with constitutive promoter, or use maize GLK gene's own promoter, and we want to see which one give better result. Okay, all right. So we use an agrobacterial mediate method for transformation using embryogenic calite. Okay, and the cultivar we use is a Japonica cultivar called Tainon 67. Okay, and then if we look at the screen on A panel, the left is a wild type, and then the next one is a transgenic plant expression G1 by ubiquitin gene, and second one by certified as promoter. They tend to be smaller after germination, and I think that the reason for that is they have smaller seeds. Okay, and then if we look at the right panel, again, the first one is wild type, the second one is G1, GLK1 from maize with its own promoter, looks more normal. However, if we express GLK2 in maize, we have some problem. Only the heterozygous plant can grow into a whole plant, but the homozygous plant for maize GLK2 gene, they don't develop into a whole plant. Eventually, they die. But if we express both of them with their own promoter, they show very strong seedling. So this is the advantage of using their own promoter. And also, if you express both of them simultaneously, confer really nice vigor in seedling development. So these are 7-day-old seedlings. After 4 months, you can see the plant morphology in the lower panel. Okay. Okay. On the left panel, you got wild type, GLK1 plant, GLK2 plant. And then on the right panel, again, you have wild type, G1 plant, G2 plant, G1, G2 plant. And you can see all the 5 transgenic lines grow much bigger than the wild type, especially when their own promoter was used to drive their expression in rice. Okay, so after we get these, we do a number of physiological study and biochemical studies. So in this case, we check gene expression. Okay, A panel show the gene expression pattern in maize. So G1 and G2 gene highly expressed in young leaf of maize plant, lower expression in mature plant, and not much in stem, root, or floret. Okay, yeah. And then, okay, but in transgenic rice, you can look at in the B panel. The G1 gene is highly expressed in leaf, stem, floret, and root in maize G1 plant. Okay, and same thing for GLK2, also highly expressed in all tissues. Okay. All right. Okay. And then if we look at the transgenic plant produced by their own promoter, okay, they tend to express at a higher level in the leaf and a little bit in the floret. Okay. Yeah. Okay. Another point I want to make is that when co-expressed or individually expressed, the rice's own GLK gene are usually reduced in expression level in the lower panel here. Okay, but the major point to say is that when both of them co-expressed, they show a coordination in expression. Okay, so this is an important point to make. Okay, then we use transcriptome to see what the gene in rice plant, in the transgenic rice plant, to express. In other words, we want to know by introducing the maize GLK gene into rice plant, what are the rice gene get affected? Okay. To our surprise, in both leaf and root, photosynthesis-related gene upregulate. These including polar, GNC, light harvesting complex of the photochemistry, but also the gene involved in abiotic responses upregulate too. These include oxylipin, phytoalexin, chitin. So basically, these data suggest that the two maize GLK gene transcriptionally promote rice photosynthesis, metabolism, and also defense response gene in a complementary manner. Okay, but generally, maize GLK1 gene act more as a transcriptional regulator, but GLK2 act more as a defense regulator. So both genes are working hand in hand in a very complementary manner. Okay, so I won't go through the detail of the data. Okay, so these are the message to take home. Okay. Okay. We also did cross section by electron microscopy examination, and we can see enhanced chloroplast development in both mesophyll cell. This is wild type, and this is a G1 using ubiquitin, G2 using certified as promoter. These three are by their own promoter. Okay. So not only they are bigger, but also they are more well developed. And we can also see more chloroplast developed in the bundle sheet cell. And usually rice bundle sheet chloroplast are relatively empty, not too many chloroplasts developed. Another feature I want to mention is that you may not be able to see it from the screen, the chloroplast developed in the transgenic plant tend to produce more thylakoid membrane, stacked thylakoid membrane. This means they have more production of NADPH, the reductant for CO2 assimilation. Okay, so this is an important point to make. Okay, and then we check chlorophyll content in leaf floret, and we also check enzyme like photosynthetic enzyme Rubisco, and also soluble protein in the leaf. As you can see, there are enhanced chlorophyll content in all transgenic plant. Okay, in all transgenic plant, increased by 18% to 39%. For the floret, you can see from the picture here in panel B, increased by 10 to 127%, especially in the ubiquitin promoter. Okay, by the ubiquitin promoter. We also show increased Rubisco content. This is the key photosynthetic enzyme in C3 plant. In the case of ubiquitin plant, 78% increase, or in the two gene, G1, G2 gene, 37%. Okay, so these are substantial increase. And of course, the whole leaf soluble protein content also increased by 21 to 28%. So therefore, we show increased leaf and floret chlorophyll protein Rubisco content, and these should increase photosynthesis in the transgenic plant. And certainly, our measurement on photosynthesis support this conclusion. You can see that in panel A, photosynthesis increased by 5 to 11% in the 5 transgenic lines, and the stomatoconductance also increased by 10 to 37%. And then the mesophyll resistance in panel C increased by 6 to 14%. And the light use efficiency increased by 4 to 16%, especially for the G1 and G2 plant. That means both maize genes are expressed in the rice, okay? Very significant, okay? We also compare their growth in the greenhouse. You can see that all transgenic lines show increased biomass by 9 to 80%. Okay, panicle number per plant increased by 2 to 37 to 124%. And then total grain weight per plant increased 2% to 70%. Okay, again, the ubiquitin promoter lead to lower germination. Okay, we also check the seed germination rate, decreased by 18%. Okay, and later I'll show you it has a smaller seeds. Okay, by this strong promoter. So if you use a strong promoter expressed in the floret, it may be causing some negative effect on seed development. Okay, and then you can look at here in panel D. This is 1,000 grain weight indication of seed size. The ubiquitin-driven G1 protein show decreased seed size. Okay, substantially. and then that may affect seed germination. Okay, so the side effect with a strong promoter could be causing decreased productivity. Okay, yeah. Okay, so in conclusion, our result demonstrate that simultaneous overexpression of both maize GLK gene under the control of their own promoter in rice can confer the most significant beneficial effect on the development of chloroplast, the expression of photosynthesis-related and stress-related gene in a complementary manner. So this in turn boosts rice photosynthesis and increase growth and grain yield by up to 70%. And of course, you would argue these are the greenhouse data. So we are doing a few trial right now. Actually, a few days ago, we harvest the G1, G2 plant, and they show a very significant increase in biomass and grain yield as well. So this is a very promising result. So another point to make is that the same approach can be extended to improve the yield of many other crops, with the potential of creating a second green revolution. So this is the end of my first lecture. Okay, let's see. I want to close the screen. Okay. Stop share. Okay. Okay, I'm back.
Okay, thank you from Maris. It's very interesting research. We hope that we can collaborate about with this research. Okay, we still have around 20 or 25 to discuss about Prof. Maurice research. If a student have a question, please raise your hand, or you can write in the chat room, or not only for student, for a college lecturer, or there is one who raised his hand is Rusmi. I'm sorry, Rusmi. It is wait. Any question? Okay. I see someone raise her hand.
Okay.
Oh, Rusmi.
Okay.
Bu Rusmi Wiyati.
Okay.
Okay, I'm sorry. Okay.
Bu.
Okay. Okay. Okay, thanks you, Bu Rusmi.
Thank you, Bu. Hello, Professor Morris. How are you? It's nice presentation you presented
fine.
before. It's really interesting research that you have presented, and because my background is from the crop protection field, I'm really curious about the defense part that you put in almost in the end of your slides, and I was quite curious about the roots development with your data. Unfortunately, you haven't had any up there, but I was really curious because it was related with the lower grain yields and grain size, and also lower seedlings with the strong promoters. But what about the roots? Sometimes smaller grain size doesn't always reflect on the root development. Am I right? But please correct me if I'm wrong. So I'm quite curious about this. Could you please extend a little bit explanation about that?
Yeah, very good point. But let me make the point first. These are grown under same size of root medium and managed equally. Everything is under the same management practice of our practice. So these are compared under this condition. So that's why we want to do the field test. Like you say, yeah, root development is very important to support the above ground development, including biomass and the grain productivity. So we need to do this. Very good point, but we haven't had a chance to do this yet. Yeah. So in the field, right now we are doing. Eventually, we probably would dig out the whole plant and check their root development. See how they. I mean these GLK gene, especially if you remember my PowerPoint. Both GLK1, GLK2 from maize also express highly in root. Okay, so this is very interesting. So the root also have these genes, but of course, if the root do not get light, they won't be green. So if we do agar culture medium, their root is green. Okay, so so they can develop into a photosynthetic capacity, but of course, underground they don't get light exposure. But in other words, they still can have stress tolerance enhancement in both root and the shoot. So the root, I expect maybe affect as well in a positive way. Okay, so this is a really good point. Thank you for raising this question. We need to do more characterization of these plants. So very good. Thank you. Yeah.
Thank you, Professor Morris. That was really interesting. Yeah,
Yeah.
and I just recall that this rice is quite what is it? Sensitive to zinc, if I'm not mistaken, zinc deficit, metals deficit in the
Sorry,
in this.
I couldn't quite follow you.
Well, yeah. Your explanation about the root development is quite what is it? Not quite. It's really interesting for the forthcoming research. I think related to the metal tolerance and all. Considering that rice development in this with wet area, it's quite sensitive to a metal toxicity, right? So yeah, thank you for your explanation, Professor Morris. Really
Okay,
nice.
okay, yeah. Okay,
Okay, thank you Bu Rusmi, or Mbak Rusmi aja, masih muda. Oke, ya. Ada pertanyaan lagi, mungkin mahasiswa bisa tanya pakai bahasa Indonesia, nanti kita yang translate. Ini ada pertanyaan dari AC, tetapi nanti ini materinya. Oke, Agam raise his hand.
Yeah,
Agam.
yeah, welcome.
Okay, Agam.
Thank you very much, Prof. Kusmiati, and thank you very much, Prof. Maurice. I would like to ask one question about biotic stresses, because you are in your slide, you are explain a little bit about biotic stresses. My question is, which one of the best plant resistant to pests or plant resistant to virus? Because there are a lot of pests, lot of insect. There are provide pathogen to plant. Which one of the best we get a plant resistant against pests or virus? And if we are developing some plant for genetic engineering, which one promoter can be used, constitutive promoter or inducible promoter? Thank you, Prof.
Okay, yeah, this is an interesting question. So you say which would be more important, resistant to virus or to other pathogen, like a bacteria or fungi? This is your question? Which one is more important? Is that
most important virus or pest insect?
yeah?
Like
This is difficult to answer because it depends on the crop. For example, in rice, the major pathogen is this leaf blight. Okay, so this is a major virus. So of course, rice also have an insect, grasshopper, problem. So you just have to check which crop you are studying. What are the major biotic stress? Insect or because of pathogen like a virus or or bacteria. For example, in banana, it's a fusarium. The bacteria is most damaging. In papaya, it's the virus, rain spot virus, cause the major problem. So you just have to focus on the particular crop, and then determine which one is more damaging, and then you put more focus on the development of resistance to that particular pathogen. Okay, so I cannot tell you just in general which pathogen is more important to control. You have to know the crop first and how they interact with pathogen. Okay, with that knowledge, you can narrow down your focus of research to do insect resistance, to raise insect resistance or pathogen, virus or fungal disease. Okay. Yeah.
Okay, thank you, Prof. And it's one of the best for using promoter constitutive or
yeah, right. The second question. Yeah, generally, if you want to raise pathogen resistance, you can use constitutive promoter. That means the defense gene can be expressed throughout the whole plant. Okay. For example, these mosaic virus certified as promoter. It's a universal promoter, constitutive promoter. It's everywhere. So the pathogen can attack every part of the plant. So if you use that promoter, your foreign gene will be expressed everywhere to defend the plant to pathogen. Yeah, very good question. Yeah, so you're thinking this good question very well. Yeah.
Okay, thank you, Prof. Maurice, and thank you, Prof. Kusmiati.
Okay, thank you, Agam. Okay. Any other question from the student or from the audience? We still have around 5 to 7 minutes. Prof. Maurice, I have one question about your transgenic rice.
Okay.
You said that the chloroplast in transgenic rice land are bigger. It is mean that the phenotype of the leaf, I mean, is it the leaf area of those transgenic rice is more wider or not?
More
the leaf area.
Huh?
The leaf area of
leaf area. Yeah.
Yeah.
There's no change in leaf size. If you look at the leaf, they are comparable in size. The leaf length, leaf width about the same, but per unit leaf area, they contain more chloroplast and more protein, more photosynthetic enzyme. So when we measure photosynthesis rate on a per unit leaf area, they have higher photosynthesis, up to 28%. Yeah.
Okay.
Yeah.
Okay. One question, Prof. Maurice. In Indonesia, the transgenic plant is still debatable.
Right.
If the breeder release the transgenic plant, it's very difficult.
Right.
How about in your country?
Still no.
Okay.
Yeah. So that's why I say, if you compare natural mutation and transgenic plant, especially transgenic plant produced by CRISPR, you
Yeah.
only change one nucleotide or three nucleotide, usually very small change.
Yeah.
That is very little change compared to chemical mutagenesis. If you use a sodium azide as a chemical mutagen, you generate a big deletion in DNA, and people are not afraid. Now we produce a transgenic plant with only a change in the nucleotide. People tend to worry. Okay. There are some argue in this aspect, but I would say, especially by CRISPR, gene editing by CRISPR, just change one nucleotide or three nucleotide. The change is rather small.
Yeah.
And then the transgene construct can be eliminated after editing. This is a nice part. So by CRISPR, your foreign gene for editing is located in one side, and the target gene for change is on the other side. Okay. After transformation in the segregation population, you can eliminate that gene, transgene, but you still have your gene edit. Okay. For example, we just demonstrate this in our transgenic rice produced by CRISPR to enhance, to change quality of starch. So maybe in the future, I can share that. Very precise. This is almost same as SMP in the natural variation. Change only by one single nucleotide. Okay, and then no transgene in the genome of the plant. Okay, but I think in Indonesia, I have a colleague many years ago, Professor Bambam Sugiharto at Jember University. He produced transgenic sugarcane tolerate to high temperature and then drought. And his cultivar is commercialized in East Java, already commercialized and planted in large scale right now. I don't know the detail of that transformation, but I think if it proves very safe to the environment, and there's no worry for the escape of that transgene, then I think maybe okay. I would not say 100% for sure. Yeah, because everybody has a concern environmentally. Okay. Yeah.
Okay. Thank you for the explanation about transgenic plant, especially about using CRISPR. Our colleagues who study in Thailand also study about CRISPR.
Okay, good.
Yeah. Okay. Any other question for the audience? We still have 1 minute before we have a break for 30 minutes. Any other question from the student? Okay. Now it's time for break. We have around 30 minutes for the break. We will start at around 10:55 or around 11 o'clock in Indonesia.
Okay.
Okay,
So
thank
30
you
minutes from now.
Yeah, 30 minutes from now. Okay, thank you from Maurice.
Okay.
We will start again in 30 minutes. Okay, thank you.
Great. Yeah, we'll come back.
Ya. Oke, kita mulai nanti 30 menit lagi ya. Silahkan, yang mau sarapan, silahkan sarapan.
Baik, Prof.
Oke.
Oke.
Oke. Mbak Oci ini tidak perlu keluar ya, Mbak Oci ya?
Tidak, Prof.
Ya.
Di Zoom aja.
Ya.
Kita mau setel video profile agro. Sudah pada lihat, tapi ya nggak apa-apa lah dilihatin lagi.
Hi, Ochi.
Yes, teacher.
Yeah. So you think the student can follow? Mhm.
Yeah, I think. But I think our student still need the basic explaining about the plant transformation. Not
Yeah.
not for plant transformation, but the modern technology for improving plant. They still need the basic explanation, such as how to get the GLK genes, how the way to get those genes, and how to insert to the rice. Yeah. How come the gene from maize go to the rice? I think they still need to
that
have the
part.
explanation about that. I think you can explain more the basic theory about the plant transformation in the second section.
Yeah, because short time for the presentation, I cannot cover everything. So that's
Yeah,
why
sure.
I just show the eventual result
Yeah.
without going through the detail of
Okay.
technique. Yeah. So yeah. Okay. So okay, I'll take a break. Oh, you have some other comment?
Yeah, actually, I think students need to know how the opportunity of C3 plants can improve to be a C4 plants, because
Right.
I remember when you give the lecture about photosynthesis when I doing my master degree, you explain that naturally plant will evolve from the C3 physiology to the C4 physiology.
C4, right.
Yeah, because there are almost C3 plants at first, but there is some plants show the intermediate
Right.
character between C3 and C4. That's
Right.
why there are some opportunity for C3 plants to improve to be C4 plants. Yeah. So C3 to C4 not kind of magic, but there are evolution theory and basic to do those kind of technology. Yeah, this is very basic. I
Right.
think.
Yeah.
And student need to
Actually,
know.
in the U.S., I spent more than 15 years study these C3, C4 intermediate plants. They are not perfect yet, not a perfect C4, but they do have enhanced photosynthetic property, reduced photorespiration. So this is what we are doing right now, because in the next lecture, you will see we are not yet to reach that specialized plant anatomy yet, because a number of genes we put into rice, we cannot make it perfect maize plant anatomy, not yet. So we are still missing in the gene identification category. We haven't find the key regulator for plant anatomy formation, but we can make it intermediate C3, C4 plant, and that has improved photosynthetic efficiency and productivity as well. Yeah. So hopefully, in the future, they can be exposed.
Totally.
Do they have plant physiology course?
Yes.
Undergraduate?
Yeah.
Yeah. Yeah. So that would help a little bit. Yeah.
Yeah. They learn about photosynthesis at the plant physiology lecture.
Right.
However, the theory of photosynthesis is just the basic theory. There are the Lie reaction and Kelvin cycle, something like that. Yeah, about the evolution of photosynthesis, and then how the variation between photosynthesis, C3, C4, I think they got the basic theory about that.
Yeah. So what I would suggest in this case, if they are interested, it's very easy to Google these days to get a Wikipedia to explain. So Wikipedia is a very good source of information. For example, there are evolution of C4, how photosynthesis, photorespiration taking place. So encourage students to go through search, to search the basic information. That would help them. These days, everybody just do Google and search for basic information.
Yes.
So
because
in this lecture, we cannot really cover everything. In such a short presentation. So
true.
Right.
Google provide everything, but depend on the person want to go to Google or not. So
That's
student, please notice the Prof. Morris School message. You guys have to search everything yang tidak ada di perkuliahan, kalian harus cari sendiri ya. Itu pesan dari Prof. Morris School. Karena yang teman-teman dapatkan misalnya di fisiologi tentang fotosintesis itu sangat dasar ya, reaksi terang, Kelvin cycle, variasi, fotosintesis, tapi ada hal-hal yang lebih dalam yang teman-teman harus cari ya, misalnya bagaimana evolusi dari fotosintesis dari C3, C4. Hampir kebanyakan tumbuhan secara alami itu C3 dan sangat sedikit sekali yang C4, tetapi ada bukti-bukti evolusi yang membuktikan bahwa ada tren perubahan evolusi dari tanaman C3 atau tumbuhan C3 menjadi C4. Nah, itulah kenapa kemudian tanaman padi ada peluang ya untuk diimprove menjadi tanaman dengan fisiologi fotosintesis C4 karena ada bukti evolusi yang membuktikan itu gitu. Ada bahwa ada beberapa tanaman yang pada awal perkembangan C3 kemudian dia menampilkan karakter intermediate yaitu karakter fisiologi fotosintesis antara C3 dan C4. Kemudian ada yang totali C4. Nah, itulah kemudian yang menjadi peluang dari ide ini gitu. I think student got a lot of insight from Prof. Morris School, and student still need to go to the literature to find more about this matter.
Right.
Thank you.
Okay, I'll take a break and come back in 20 minutes or so. Okay. See you later.
See you. Prof, course kita mau share video profil dulu ya, Prof, untuk mengisi break.
Program Studi Agroekoteknologi merupakan salah satu program studi yang ada dalam Departemen Pertanian, Fakultas Peternakan dan Pertanian Universitas Diponegoro.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Perkenalkan, saya Insinyur Karno MAPPL SC PhD selaku Ketua Program Studi Agroekoteknologi Fakultas Peternakan dan Pertanian Universitas Diponegoro. Saya akan menyampaikan visi dan misi dari Program Studi Agroekoteknologi. Visi dari Program Studi Agroekoteknologi FPPUNDIP adalah bahwa pada tahun 2022 menjadi salah satu pusat unggulan dan rujukan ilmu pengetahuan dan agroekoteknologi kawasan tropis untuk mendukung pembangunan nasional dan peningkatan kesejahteraan masyarakat yang berkelanjutan. Sedangkan misi dari program studi S1 Agroekoteknologi FPPUNDIP adalah yang pertama, menyelenggarakan dan mengembangkan kegiatan pendidikan, penelitian, dan pengabdian kepada masyarakat, serta pengembangan kemitraan dan kerjasama yang berkelanjutan dengan mengutamakan pemanfaatan secara lestari atas tumbuhan atau tanaman dan sumber daya alam lokal. Misi yang kedua adalah menyelenggarakan manajemen atau tata kelola penyelenggaraan pendidikan tinggi, khususnya prodi yang baik, transparan, akuntabel, bertanggung jawab, dan adil. Good Study Program Governance. Saat ini, program studi agroekoteknologi FPPUNDIP memiliki 15 dosen aktif dan 5 diantaranya bergelar profesor. Sejak berdiri pada tahun 2013 hingga saat ini, program studi agroekoteknologi sudah meluluskan lebih dari 200 mahasiswa. Lulusan program studi agroekoteknologi Undip diharapkan memiliki profil kompetensi sebagai pelaku di bidang pertanian, manajer, pengusaha, peneliti, dan komunikator. Saat ini alumni prodi agroekoteknologi tersebar di berbagai macam bidang kerja. Ada yang bekerja sebagai pegawai swasta, pegawai pemerintahan, wirausaha di bidang pertanian, dan melanjutkan studi S2 sesuai minat masing-masing. Program studi agroekoteknologi FPPUNDIP terus berupaya menjadi bagian pembangunan pertanian di Indonesia. Wassalamualaikum warahmatullahi wabarakatuh.
Kegiatan akademik di program studi agroekoteknologi didukung oleh sarana prasarana yang tersedia, seperti perpustakaan, pengelolaan sistem informasi, ruang kuliah yang dilengkapi dengan komputer, LCD dan AC, gedung pusat kegiatan mahasiswa atau PKM, serta laboratorium untuk kegiatan praktikum dan penelitian. Selain dilaksanakan di laboratorium, kegiatan praktikum dan penelitian dilakukan di greenhouse, screenhouse, dan agrotechnopark yang merupakan kebun Fakultas Peternakan dan Pertanian Universitas Diponegoro. Fasilitas pendukung lainnya yang disediakan untuk memberikan rasa aman dan nyaman seperti musola, kantin, dan gazebo. Untuk meningkatkan SDM mahasiswa, program studi agroekoteknologi dan Departemen Pertanian memfasilitasi mahasiswa dalam suatu wadah, yaitu Himpunan Mahasiswa Departemen Pertanian atau yang lebih dikenal dengan HMD Pertanian.
Perkenalkan, saya Arif, founder dan CEO dari CV Ayunani. Kesempatan belajar di agroekoteknologi Undip menjadi bekal penting bagi kami dalam membangun dan mengembangkan usaha kami. Tidak hanya belajar mengenai tanaman, tapi bagaimana kami memandang secara lebih luas dunia pertanian, sehingga kami bisa ikut mengambil peran sekaligus peluang dengan berwirausaha di bidang pertanian ini. Yuk, usahanya anak muda bergotong royong membangun pertanian Indonesia.
Halo, saya Nyoman, Agroekoteknologi 2013. Kini saya bekerja di Astra Group, PT Astra Agro Lestari Perkebunan Kelapa Sawit, penempatan di Riau. Sebagai alumni, saya merasa pendidikan di Universitas Diponegoro, khususnya Agroekoteknologi, sangat bermanfaat untuk saya bekerja sekarang. Dan untuk kalian semua, selain pintar di kelas, jangan lupa berorganisasi. Dah.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Muhammad Rizky Agam. Saya merupakan alumni dari Agroekoteknologi Universitas Diponegoro. Alhamdulillah pada kesempatan ini saya berkesempatan untuk melanjutkan studi di jenjang magister pada konsentrasi pemulihan dan bioteknologi tanaman di Institut Pertanian Bogor. Alhamdulillah semasa kuliah di satu agroekoteknologi ini saya mendapatkan banyak pengalaman dan juga mendapatkan banyak ilmu yang diberikan oleh dosen di sini, terutama di bidang pemulihan dan juga bioteknologi tanaman. Alhamdulillah saya berkesempatan juga untuk menjadi asisten dosen serta dipercayai juga untuk menjalankan proyek penelitian yang berkaitan dengan pemulihan tanaman. Mungkin itu yang bisa saya sampaikan. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Agroekoteknologi adalah suatu program studi yang mempelajari tentang ilmu pertanian. Selain itu, di sini kami juga belajar mengenai berbagai macam soft skill yang membuat kami lebih berani untuk terjun di masyarakat.
Di program studi agroekoteknologi, saya bertemu dan diajar oleh dosen-dosen yang ahli di bidangnya. Dan saya juga bertemu dengan teman-teman dari berbagai macam daerah, sehingga saya lebih mengerti apa artinya Bhinneka Tunggal Ika.
Halo teman-teman, mari bergabung. Di agroteknologi.
Ya, selanjutnya ini ada video profil Fakultas Peternakan dan Pertanian Undip. Bapak Ibu peserta yang kebetulan dari universitas di luar Undip mungkin bisa menyaksikan.
Welcome to the Faculty of Animal and Agricultural Science, Diponegoro University. We have centralized one campus in Tembalang, Semarang, Central Java, Indonesia.
Semua, Perkenalkan, saya Dr. Insinyur Bambang Walyadi Eko Prasetyono, MS MAGRSC IPU, selaku Dekan Fakultas Peternakan dan Pertanian Universitas Diponegoro. Fakultas Peternakan dan Pertanian Universitas Diponegoro atau lebih dikenal dengan FPPUNDIP merupakan salah satu fakultas yang ada di Universitas Diponegoro, Semarang. Pada Fakultas Peternakan dan Pertanian Universitas Diponegoro adalah menjadi fakultas unggul bertaraf internasional bidang iptek peternakan dan pertanian kawasan tropis tahun 2030. Sedangkan misi Fakultas Peternakan dan Pertanian Undip yaitu yang pertama, mengembangkan program pendidikan unggul bertaraf internasional bidang peternakan dan pertanian tropis untuk menghasilkan lulusan yang kompetitif. Nasional bidang peternakan dan pertanian tropis untuk mendukung peningkatan daya saing bangsa. Dan yang tiga adalah mengembangkan layanan unggul bertaraf internasional bidang peternakan dan pertanian tropis untuk mendukung peningkatan kesejahteraan masyarakat. Dan yang keempat adalah mengembangkan tata kelola fakultas yang efisien, efektif, akuntabel, dan transparan.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, saya Dr. Insinyur Limbang Gustiawan Nuswantara, SPTMP IPU, yang pada saat sebagai Wakil Dekan Bidang Akademik dan Kemahasiswaan. Terus saya sampaikan di sini bahwa Fakultas Peternakan dan Pertanian Undip itu didukung oleh 103 orang dosen yang terdiri dari 24 orang dosen memiliki jabatan guru besar atau profesor, 46 orang dosen memiliki jabatan dosen, 11 orang dosen memiliki jabatan asisten ahli. Sampai saat ini, Fakultas Peternakan dan Pertanian Undip memiliki jumlah mahasiswa aktif sekitar 244 orang yang terdiri dari 1.119 orang asisten ahli madya, kan 6.587 orang sarjana dari program studi peternakan, 325 orang sarjana teknologi pangan, 225 orang sarjana agribisnis, 196 orang sarjana pertanian dari program studi agroekoteknologi.
Agroteknopark Fakultas Peternakan dan Pertanian Undip dibangun dalam rangka untuk fasilitas pembelajaran di Fakultas Peternakan dan Pertanian, di mana di situ ada kandang ayam close house dengan sistem modern dengan kapasitas 33.000, ada kebun buah, ada lab untuk kewirausahaan, kemudian ada screen house, dan lain-lain. Fasilitas-fasilitas ini dibangun untuk menunjang pembelajaran, yang sisa hasil praktikumnya merupakan RGE bagi fakultas, di mana RGE yang dapat dihasilkan dari kegiatan ini cukup banyak sehingga dapat digunakan untuk perbaikan laboratorium yang ada di fakultas.
Kita lihat di belakang saya ini ada berbagai fasilitas pendidikan yang terkait dengan bidang peternakan dan pertanian. Kami dari Fakultas Pertanian memiliki beberapa fasilitas laboratorium, yaitu laboratorium ternak potong dan laboratorium ternak perah. Di sana ada berbagai ternak, baik itu sapi semental, sapi Brahman cross, kemudian sapi-sapi lokal ada, kemudian sapi-sapi silangan yang lain juga ada. Kemudian kita punya juga sapi perah ya, jenis Friesian Holstein yang ada di belakang layar ini. Kemudian ada lagi nanti di sebelah sana ada fasilitas kita punya untuk praktek dan riset untuk kambing juga, baik itu kambing potong maupun kambing perah. Juga ada nanti sebelahnya lagi ada laboratorium ayam potong, ya, ayam broiler, dan juga ada ayam petelur kita juga memiliki. Dan ada lagi ternak, kita memiliki rusa yang ada di depan maupun yang ada di belakang di kanat. Itu ada fasilitas untuk laboratorium rusa. Kemudian kita juga punya untuk keperluan pakannya ada mini feed mill atau pabrik pakan mini yang dipakai untuk penelitian-penelitian dan praktek-praktek untuk adik-adik mahasiswa. Dan juga setiap hari kita produksi untuk memenuhi kebutuhan pakan sendiri, sehingga pakannya itu tidak disuplai dari luar, tapi kita sudah membuat pakan konsentrat sendiri untuk pemenuhan ternak-ternak yang ada di lingkungan Fakultas Pertanian dan Pertanian Undip.
Menjadi bagian dari Fakultas Peternakan dan Pertanian Universitas Diponegoro merupakan salah satu kebanggaan bagi kami.
Di sini kita belajar banyak hal mengenai ilmu dalam bidang peternakan dan juga ilmu di bidang pertanian.
Bertemu dengan teman seperjuangan
Untuk memajukan peternakan dan pertanian di masa yang akan datang.
Mari bergabung bersama kami dan menjadi bagian dari keluarga besar Fakultas Peternakan dan Pertanian.
FPP Jaya, Undip Jaya.
Okay, good afternoon, Prof. Maurice.
Yes.
Okay, it's okay if we continue the lecture.
Okay.
Okay, for the next session, we will discuss about, it's very interesting topic is about production of C4 rice. So I hope all the students already know rice is C3, but Professor Morris change from C3 to C4 rice. Okay, time is yours, Prof. Morris.
Thank you.
Bu Oji, itu apa bahasanya? Bahasa hurufnya huruf Thailand.
Yeah.
I have some
China.
problem.
Okay.
Just a second. Okay. Try to find my slides.
Yeah. You can open your PPT files.
Okay.
Okay.
Yeah. Successfully displayed.
Okay. Okay. I'm all set. Can you hear me?
Yeah, it's very clear.
Okay. Okay. I hope everybody is back. In this third part of my presentation, I want to talk about sustainable agriculture in facing climate change. The central theme of this study is production of C4 rice. Okay. Since my graduate study, I work on C4 photosynthesis. Early days in my PhD study, I work on photosynthetic characters in C4 plant, including the C4 photosynthetic pathway, the enzyme involved in the pathway, and then do all kinds of assays. And then after I graduate, I begin to work on some intermediate species between C3 and C4 plant. And then about 20 years ago, I began to work on production of C4 rice. Everybody by now should know rice is a C3 crop, has an inferior photosynthetic character and lower productivity. So I'm thinking of maybe it's possible to convert rice plant from a C3 to a C4 plant. So that's the overall of my goal, starting 20 years ago, because the gene responsible for the C4 pathway has been isolated, well characterized. So about 20 years ago, I first introduced maize C4 enzyme called PP carboxylase and introduced into transgenic rice, and with a big success. And then, of course, we know there are many genes involved in C4 photosynthesis. So in the last 20 years, we begin to do more transformation with in rice with more maize genes. Okay, so this is how it begins. So this work almost built from the last 20 years, and we are only making a small step in progress. The reason is very simple, because there's a food shortage that represent a great challenge to meet the population demand. If you look at the population increase in 17th century, it's less than 1 billion people on Earth. 18th century, about 1 billion. And then to 1927, 2 billion, 1960, 3 billion. There's a very rapid increase in population. Almost every 10 years, there's 1 billion people increase. And right now we are closing to 8 billion. And then it's project 9 billion by 2048. So we need to produce at least 40 to 60% more food by 2050, which is only 30 years away. So this is a population demand for productivity increase. The other complication is that there's a deterioration of our cultivated land. And then this drastic climate change can further reduce crop productivity or crop yield. So, and if you look at more statistics as shown in this slide, from 2006, the rate of crop yield increase is slower than the rate of population increase. Here, as you can see, excuse me, this is a population increase, and our current crop increase, if it stay the same pace, you will see a bigger and bigger gap between the rate of increase in productivity and the rate of increase in human population. And this is the worst in a developing country, okay? Because the total cereal production increase is slowing down in the last 10 years, okay? Yeah, so there's a need to largely increase the yield increase, okay? Everybody knows the Green Revolution, which took place in the 1967 to 1992. Basically, it's adoption of a semi-dwarf variety in rice and in wheat. Okay, and this semi-dwarf variety is because they have a mutation in gibberellin production, so they become shorter. So basically, it's about a new architecture for the crop. However, the benefit of a green revolution has been exhausted. So we need to use another approach to increase these crops productivity. And of course, you know, wheat is also a C3 crop besides rice. Okay. And the limitation of conventional crop breeding is that it's very ineffective, very time consuming. Okay. Another problem is that there's a barrier in gene transfer from different genetic sources. The conventional breeding usually take two variety and you do hybridize and then screen for higher productivity or resistance to pathogen. Okay. If the genetic distance between the two cultivar is very limited, the improvement is also very limited. Okay, so this is another barrier, what I say in gene function. Okay, so other mean, as you also know, hybrid crop has been used to improve a number of crops. The key point for hybrid crop is that they have to have wide genetic distance. So the hybrid vigor is stronger. If you take a two genetically closely related cultivar and you do hybridization, you won't expect to get any new trait at all because they are so similar in genetic background. Okay, so these are the limitations with conventional crop breeding. Okay, so we can use modern technology, especially in gene discovery and then transformation for crop improvement. As I mentioned in my first part of lecture, these could be chemical genetic mutagenesis to find the new gene function. We can use a QTL to stack the useful gene to raise productivity. And then we can make this process more efficient based on marker assisted breeding. Okay. And then in my talk, I will show you how effective transcriptome analysis can be used to identify important gene with new functions. Okay. And then in the future, genetic transformation can be done by this new technology called CRISPR. Okay. Yeah. So I mentioned this earlier. I won't go through the detail here. Okay. About 30 years ago, we have the first conference at Erie, Las Palmas, Philippines. The central theme is that we have to redesign rice photosynthesis to increase yield. Okay, so this is a publication by Erie about almost 31 years ago. On the cover page, you can see that the light use efficiency for rice crop is only 3%, but it's about 5% in corn or wheat. Okay, so the conclusion from that conference is that rice yields are fast approaching a theoretical limit set by its efficiency in harvesting sunlight and using this energy to make carbohydrate. Okay, so to feed the world, rice may have to be re-engineered at the biochemical level. The only way to increase yields and reduce nitrogen fertilizer is to increase photosynthetic efficiency. Okay, so these are the major message from this conference. Okay, photosynthesis is more efficient in C4 plant than in C3 plant or C3 photosynthesis, C4 photosynthesis. C3 photosynthesis suffer from photorespiration. Okay, this is a wasteful process which reduce plant productivity by 30 to 40%. Okay, simply because they have this photorespiratory process. Okay, and then unfortunately, many major conventional crops, rice, wheat, soybean, potato, many vegetables are all C3 plant. And of course, we know that during evolution, C4 plant evolved to overcome photorespiration, and they are more efficient. We know C4 plant include some of the important productive crops like maize, sorghum, sugarcane, and they are more heat, drought tolerant, have a higher carbon, nitrogen, sulfate use efficiency. So about 30 years ago, I set the goal that I want to introduce C4 photosynthesis into C3 crop to boost their productivity. Of course, I use rice as a model plant to do this, okay? And just quickly to go over the photosynthetic and productivity of C4 versus C3 plant. In our paddy, because rice is a C3 crop, there's another wheat. It's called Echinochlora cruscali. This is a weedy species adapted to the paddy field. If you look at the photosynthetic temperature response, yeah, rice has lower photosynthetic rate. At the optimum temperature, 30 degrees, it's about 30 units of photosynthesis. But if you look at this C4 weedy species, it has a much higher photosynthetic rate throughout the whole temperature spectrum, especially at higher temperature. It can tolerate to heat as high as 40 degrees, okay? And then rice has a decreased photosynthetic rate at a higher temperature. So this is a very drastic difference in photosynthetic capacity between C4 and C3 rice. If you look at the nitrogen use efficiency, you grow maize, sorghum, and rice or wheat at different nitrogen content in the soil with different nitrogen or fertilizer in the soil. And you look at the rate of photosynthesis in the leaf, you can see maize show the highest efficiency. There's a rapid increase in photosynthetic rate with increasing nitrogen content. And next, you can see also drastic increase in sorghum, but at a less efficient degree. And in significant contrast, you can see rice and wheat show a very slow increase in photosynthetic capacity in response to increasing nitrogen application in the soil. You see, at this high level nitrogen, its assimilation rate still lower than maize and sorghum. Okay, so you can see the disadvantage of a C3 crop. Okay. So the question is why C4 photosynthesis is more efficient or C4 plant more productive? There are two major factors. One at the structural level, because C4 plant has this specialized crunch leaf anatomy, which increased the nitrogen sulfate nitrogen fixation efficiency. Okay. And then the second feature is that C4 plant have a specialized C4 pathway of photosynthesis, which is highly efficient in carboxylation and decarboxylation. And the purpose of these two reactions is to concentrate CO2 at the site of final fixation and suppress photorespiration. So the key is C4 pathway can raise CO2 concentration in the cell and then suppress photorespiration. Okay, so there's no apparent photorespiration in C4 photosynthesis. If you put the C4 plant in a closed container, it will fix CO2, all CO2 in the container, in the closed system. Okay, but if you keep a C3 plant in a closed chamber, CO2 will be fixed until about 50 parts per million and then stay steady state at there. So you can see the major difference in C4 and C3 photosynthesis. Okay, so here you can see the drastic difference between leaf cross-section of rice versus maize. In a C3 rice, this mesophyll cell are the major photosynthetic cell. So let's differentiate a bundle sheet cell. This vascular here, they are relatively empty without too many chloroplasts. Okay, so in my last talk, I show you that by overexpression GLK1 and K2, we can increase chloroplast development in the bundle sheet cell of rice. But anyway, you can see between two vascular, there are 7 to 8 mesophyll cells. So this is long distance. When the photosynthetic product is produced in mesophyll cell, they have to cross 4 or 5 cells to transport to vascular cell. But in great contrast, you can see there are only 2 mesophyll cells between 2 vascular bundles. And then this bundle sheet cell are well differentiated with chloroplast, mitochondria, peroxisome. So this is really a significant difference. Okay, and then in maize leaf, all the photosynthetic product can be efficiently transported from mesophyll to bundle sheet cell, and then final carbohydrate can be transported out through the vascular, the phloem tissue. Okay, yeah, and so the major difference is the high vein density here. Within the same distance, you have very dense mesophyll bundle sheet cell. Yeah, in contrast to rice, low vein density. Okay, so these are two major difference between C3 leaf and C4 leaf. High vein density, and then well-developed bundle sheet cell. And then these two cell types coordinate in the C4 pathway of photosynthesis and make the CO2 utilization very efficient. Okay. Okay. Go back to the basic photosynthesis as you learn from your plant physiology courses. In the C3 plant, the major photosynthetic enzyme is called rubisco, RUBP carboxylase and oxygenase. Okay, so this is a bifunctional enzyme. It has a carboxylation to produce a net photosynthesis, but it also can go through oxygenation to produce photorespiration, and the product is CO2, and that is released to ambient. So in this reaction, when RUBP incorporate with CO2, it produces 3 PGA, 2 molecules. So this is net photosynthesis. However, if RUBP react with oxygen, it goes through oxygenation, only produce 3 PGA, one molecule. But the other product called 2-phosphoglycerate, this is the substrate for photorespiration to release CO2. So you see, on one hand, C3 prime try to fix CO2 into carbohydrate, but when they react with the oxygen, they produce a wasteful reaction called photorespiration and release CO2. So this is a very wasteful process. And this reaction of photorespiration takes place in 3 organelles in a cell. Okay, this is chloroplast. So Rubisco can react with oxygen, as I mentioned, produce a 2-PGA, 2-phosphoglycerate. And this goes through peroxisome and then mitochondria. Eventually, CO2 is released in mitochondria. And this is a photorespiratory product. Okay, yeah. Oh, sorry. Hey, wait a minute. Okay, sorry. Oh, I go back. Okay, so and then this is wasteful photorespiration CO2 released to ambient of the leaf. And then you may ask, can we engineer Rubisco to eliminate oxygenase reaction and photorespiration? The answer is no. This has been tried a few times in the past 30 years ago. They find out that both CO2 and oxygen are competing for the same binding site on Rubisco, same binding site, in a comparative manner. So high CO2 suppress photorespiration, and high oxygen favor photorespiration, as you can see from this reaction. And also high temperature promote photorespiration. And so in consequence, photorespiration reduced photosynthesis and crop productivity in C3 plant by 35 to 40%. And during evolution of plant, C4 photosynthesis evolved like 10 million years ago to overcome this problem. Because when C4 plant evolve in, usually evolve in a dry, hot area where photorespiration would be very high. So C3 plant cannot survive in that climate. Only when they have C4 photosynthesis, they can adapt to high light, high temperature, and very dry condition. Okay, so that's a key advantage of C4 photosynthesis. So let's look at the C4 pathway. There are 3 subtypes. It's a little bit complicated, but let's look at the model in C4 plant, the C4 pathway in maize, C4 plant maize. When you have ambient CO2 level around 415 parts per million right now, CO2 will first enter the leaf through stomate, and then CO2 is converted to bicarbonate in the mesophyll cell. This is the outside layer. This carbonic anhydrase hydrates CO2 to bicarbonate. This is the substrate for phosphoenolpyruvate carboxylase, the PEPC, okay, carboxylase. And the product is a C4 compound called oxaloacetate. This is 4 compound. So it's called C4 prime because the initial product of C4 photosynthesis has a 4 carbons. And this oxaloacetate is a very short-lived compound. So it enters the chloroplast of mesophyll cell and then converted to malate. Malate is more stable C4 acid. and then mainly serve as a shuttle compound, go through the plasma desmata, the tiny hole between mesophyll and bundle sheath cell, and then enter the chloroplast and release CO2 and then the product pyruvate by this enzyme called decarboxylation, NADP melee enzyme. And then the product pyruvate shuttle back to mesophyll chloroplast and converted back to PEP. Okay, this is mediated by the enzyme called PPDK, pyruvate PI dikinase. So you can see the key C4 pathway enzymes are carbonic anhydrase, PEP carboxylase, MDH melee dehydrogenase, NADP melee enzyme, and then PPDK. So this pathway run at very fast pace and release high CO2 inside the bundle sheath chloroplast, where you have the C3 cycle. Rubisco is located here. So I mentioned earlier that when you have high CO2, it can compete oxygen out. So there's no oxygenase reaction. So no photorespiration. So you can see that CO2 outside the ambient, outside the leaf in the ambient is 415 ppm, but with this C4 pathway, the CO2 released inside the bundle sheath cell is about 2000 ppm. So there's very little photorespiration. The oxygenase is low. So this is a major advantage of C4 plant. And actually in horticulture science, people use this advantage because in the horticulture setting in the greenhouse, they grow C3 crop. They can inject CO2 into the greenhouse and raise the productivity of C3 crop in the greenhouse. And same principle, okay? But here, you don't need to inject CO2. The C4 pathway will do this work for you, okay? So we set our major goal or objective first, to install a functional C4 pathway in the C3 leaf, like a rice. We may have C3, C4 intermediate rice with one cell. Okay, C4 plant has a 2 cell, and this is a long-term goal to make a C4 rice with the 2 photosynthetic cell. Okay, because it's probably easier to make a C3, C4 intermediate rice using 1 mesophyll cell, because this type of photosynthesis occur in nature. I mentioned that I studied some C3, C4 intermediate species in nature. Okay, they don't have a perfect Kranz anatomy or C4 pathway. They have partial C4 photosynthesis. So maybe without change the structure of rice leaf, just use 1 mesophyll cell, we can introduce the C4 pathway and get some enhancement in photosynthetic activity. Okay, but eventually, for more effective C4 rice would be needed with a 2 photosynthetic cell. Okay, so as I mentioned, our first goal is creation of a C3, C4 rice, intermediate rice, with only 1 mesophyll cell. No change in Kranz anatomy. And later we want to introduce Kranz anatomy. Okay, so what are the genes or enzyme we want to put into a C4 mesophyll cell? Okay, so we try to decrease CO2, increase, sorry, we want to enhance CO2 fixation in the C3 mesophyll cell. If you look at the ancestral C3 plant, of course, one cell type, like a photosynthetic bacteria, they have this bicarbonate pump, okay, because in water, a lot of CO2 under alkaline condition occur in bicarbonate form, okay, bicarbonate. And this bicarbonate is a substrate for PEPC in C4 plant. So if you can take this bicarbonate pump gene, this is on membrane, plasma membrane, okay? And then also in aquatic plant, few aquatic plant, again, they have only one cell, mesophyll cell. They have bicarbonate pump too, to better survive the low CO2, high bicarbonate water. Now in land plant, we have C3, as I mentioned. Interestingly, 90% of the plant species on Earth belong to C3, and they are distributed mainly in a temperate zone. And C4 plant with a two photosynthetic cell, the mesophyll cell, the bundle sheath cell, they only account for 3% of plant species on Earth, and they mainly tropical in nature. And of course, you know, there are chem plant like a cactus. They have one cell type. They account for 5% of the plant species, and they are distributed in arid. A chem plant and C4 plant almost share the same biochemical pathway, but chem plants separate the biochemical reaction, carboxylation, decarboxylation, in a day, night, in a night and day separation. So this is a temporal separation of C4 pathway. And in C4 plant, this is special because the C4 pathway separate into two cells to operate. Okay, and then in nature, there are C3 and C4 intermediates, as I mentioned. They have only one cell, mesophyll cell only, but they have some C4 function. So this type, C3, C4 intermediate, is our first goal to achieve in rice. Okay. And then again, every time you try to do genetic engineering, the first question is which promoter to use, the constitutive or specific promoter. And then the other question is a site of insertion in the host genome. When you introduce foreign gene into a host plant, usually you cannot predict where the gene will be inserted into the genome, in the host genome, okay? Okay, and the other question is, where would be the site of expression in plant? So this again is related to promoter, okay? And then probably depending on the insertion site, you can have a different expression level, okay? We have many cases, same gene, same transformation, but their expression level can differ by 100-fold, okay? Because the site of insertion maybe regulate the expression of foreign gene, okay? Basically, I would say we try to use their own promoter. I mean, if we take the maize gene, we like use their own promoter to express in the right cell, right place, okay, right tissue, right cell, okay, yeah. So as I mentioned, I started almost 32 years ago, yeah. We published this first paper in Nature Biotech, 1999. So this is the first breakthrough with a high level expression of maize phosphoenolpyruvate carboxylase in transgenic rice. Okay, and as I mentioned, in this study, we put this maize gene with their own promoter and had a very high expression level in rice, and there is no side effect. It enhances photosynthesis very well. Okay. Then the second gene I put into rice is called NADP malate enzyme. So PPC is a carboxylation enzyme, produce a C4 acid. And then the next step I want to do is to do decarboxylation enzyme called NADP malate enzyme. However, we find a very shocking result. High level expression of the gene either from rice NADP malate enzyme or maize cDNA. The higher the expression, the lower, the less vigorous of the citrin. Okay, yeah. This just show you the expression level of NADP malate enzyme. Okay, here. When you have a transgenic plant, this is wild type photosynthesis. With increasing light, photosynthesis increase to almost half sunlight here. But our transgenic plant with a high level expression could not respond to high light. Actually, higher light cause photo inhibition. Okay, you see, this is wild type leaf. And then these two are transgenic with a high level of NADP malate enzyme, especially from maize. Okay, these are transgenic plants. When they have higher level expression, the worse the plant look like. So the problem is this. When you express this enzyme in okay, let me say this enzyme in C4 plant, like a maize first. In maize, if you do a cross section of the leaf, this is a mesophyll cell, mesophyll cell, mesophyll cell. This is bundle cell. You can see drastic difference in structure. This is a thylakoid, stacked thylakoid for NADPH production. And this is unstacked region for ATP formation. This is a bundle-shaped chloroplast, no stack, unstacked. So they are mainly for ATP formation. Very little NADPH, okay, production. So when you introduce NADP malate enzyme, it would produce NADPH by this reaction. Okay. But remember, in rice, they all look like this. Okay, so when you introduce this enzyme into here, this enzyme will compete for NADP to produce NADPH. So here, they run out of NADP, so produce an oxidative mechanism. Okay, so they cannot do well. So that's why when you have high level expression, the plant look terrible. Okay. So because of this problem, we have to think of an alternative mechanism for decarboxylation of a C4 acid. Okay. So in the last 15 years, we pretty much use this model for our engineering of C3, C4 rice. So only one cell type in a mesophyll cell. CO2 in the intercellular space can be in CO2 form or bicarbonate form. Usually pH 7 major form is a CO2. So normally CO2 diffuse in. Okay. Some bicarbonate, we want to use the cyanobacteria ICTP bicarbonate pump to send in. So there's no need for CO2 to convert to bicarbonate through carbonic anhydrase. But both form we want it. So we want to introduce this bicarbonate pump. We also want to introduce carbon anhydrase to convert CO2 to bicarbonate. And this is the substrate for maize PPC carboxylase. So when we produce oxaloacetate, instead of a reduced to malate, we intend to have OAA transport into the chloroplast and use another decarboxylase called PEP carboxykinase. This is from another C4 plant. Directly reduce OAA, decarboxylate OAA to CO2 and PEP. You see, this is a product PEP can be shuttled back to the cytosol for this PEP carboxylation. So we hope this a minimum of 1, 2, 3, 4, or 5 gene can have functional C4 pathway. Maybe limited, but it's a functional. Okay. The other enzyme is called pyruvate PI dikinase. It can convert pyruvate to PEP. Okay, using ATP. So these are the 5 major enzymes we want to put into a rice mesophyll cell. Okay, so let's see what happened. So far, we have transgenic rice individually expression ICTB or PEP carboxykinase K1 and high PEPC, and then combination of 2 genes I and P, IP5, IP3, and then PK means PEPC kinase, PK3K1, IPK1, IPK2. These are 3 genes, 3 genes. Okay, and this 2 gene, 1 gene, and you can see that transgenic with a high level of PEP carboxylase can have a high rate of photosynthesis, and also IP combination of 2 genes may be still beneficial. Okay, but the other one, when you have a K PEP carboxykinase, they don't do so well. Okay, the reason for that is because when you overexpress K PEP carboxykinase, you make the chloroplast less stacking, more loose structure in the chloroplast structure. This is wild type, very well organized. Stacking region, little non-stacking region, but in K1, you can see very loose cytosol structure. This is not good for NADPH production, okay, less efficient. And then PK1, K plus IP, same thing. You see more loose cytosol membrane. And when K2, that means this K expression level is much higher. You can see more loose cytosol membrane. Okay, so this is a disadvantage of using PEP carboxykinase in these combination, because they are less efficient for NADPH production. Okay, but to our surprise, when we have a right combination, we don't have this problem at all. So very lucky. So you have to try different combination of C4 gene in order to see how they affect the plant structure function. Okay, and so luckily another construct we have with only 2 genes, carbon anhydrase and carboxykinase. You can see our plant much healthier, and they don't have this problem. They have higher photosynthesis, about 11% higher. Okay, and their CO2 compensation point, this is a measure of photorespiration, reduced by 5%. Okay, so that means these 2 enzymes, just a C and K, carbon anhydrase and PEP carboxykinase, it can reduce photorespiration. It helps the C4 function in this transgenic rice plant. So we are very happy to find this in this case. With a C and K, we don't have a structural problem, and they have C4 function by reducing photorespiration and have a higher photosynthetic rate. Okay, this show you the CK plant, the anatomy of CK plant. You can see this is stacking region. This is amplified magnification. Very beautiful stack. Very little loose cytochrome. Very little. Okay. Yeah. So CK transgenic rice did not show any significant change in chloroplast development, photosynthesis, and growth. So coordinate expression of these two biochemical steps can overcome the negative effect on chloroplast structure. So now we quickly make, we take an IP transgenic plant with 2 genes, with another transgenic plant with C and K, I and P. We make a hybridization, conventional hybridization, and then we screen for homozygous plant and for further characterization. Yeah, and so we also do field trial in Taichung. This is my colleague help me. This is transgenic crop. We were joking, our transgenic rice plant, especially with a PP carboxylase gene, they grow rapidly and form a very dense population. So the mother bird prefer our transgenic rice field to lay their egg. Okay. We also do photosynthesis measurement, and some of the transgenic plant, especially with PEP carboxylase, they have higher photosynthesis rate. Okay. For example, here, this is not photosynthesis rate. This is the grain weight. Okay. You can see that the increase in grain yield in ICTB transgenic plant almost 34%. Okay, K1 is less significant. And then in another experiment, we compare wild type with a PEPC transgenic and IP transgenic and IPK2. You can see that the K2 high level expression make them less productive, lower grain weight. But with PPC along two different plants with two different expression level, they can increase 8 to 12%. And then one with I and P, they can increase up to 40%. So this is rather significant. Okay. Of course, I mentioned that our final goal is to engineer rice leaf to have crunch anatomy. But the key is we have to know what are the regulatory gene or the network to convert the C3 leaf to a C4 leaf. Okay, this occurred naturally through evolution million years ago. So can we duplicate this process through genetic engineering? Okay, so luckily I find a collaborator, Professor Lee. He's from Chicago. He's an evolutionist, also a very keen molecular biologist. So we team up to understand the regulatory network that control the development of this crunch anatomy. Okay, so what we do is we use a comparative transcriptome analysis to study the development of C4 leaf and try to dissect the underlying regulatory network for crunch anatomy. We study cell-specific regulation, developmental regulation in light and dark, tissue-specific regulation, like in husk leaf and foliar leaf, because in maize, foliar leaf has a chromosomal. Husk leaf does not have chromosomal. And so in a developmental regulation, we germinate maize seeds. And then during germination and leaf development, we take sample every 6 hours until maturity. And we look at the gene expression profile and try to narrow down which genes are involved in chromosomal. Okay. And so you want to ask, so how do you know these transcription factor you identify are involved in chromosomal? Luckily, previous study has shown that 2 transcription factors called SHR, SCR, they are directly involved in vascular development. So this is good marker for us to use in leaf development. Okay, yeah. And so, for example, SHR has a 2 peak of expression after germination, 30 hours and then 56 hours. And SHR a little bit later, okay, about 60 hours. Okay, so these can serve as a marker for vascular development. Okay, and then we look at other genes. For example, GLK1, GLK2. GLK1 mainly expressed in mesophyll cell. GLK2 mainly expressed in bundle cell. You see, they start to express every day. Interestingly, they express in the night and decrease in the day, and then again in the night for chloroplast development. This is really a surprise to us. And same thing for G2. So basically, after 3 days, 3 or 4 days, they begin to express, and then chlorophyll begin to accumulate in maize leaf. Okay, yeah. So using this approach, I won't go through a lot of detail. I just want to show you that the expression of these genes, like SCR, SHR in rice leaf and maize leaf, are very different. You see, OS SCR in rice peak 3 days and then decrease all the way. And then the other gene also decrease right after germination. But in maize, it increased after 30 some hours, 50 hours. Okay, so very different. So that means these are actually involved in plant anatomy development in C4 plant. Okay, and this just show you a different expression pattern of some of the transcription factor. I won't go through the detail. You can see those important for plant anatomy develop before 3 days in maize, but they all decrease in rice. Very different. Yeah. Okay. And so finally, we come up with this regulatory mechanism depending on the time. So like a TERF, GRF, this growth regulator factor, very important. They express very early stage of maize leaf development. And then there are ARF, auxin response factors, WAKI gene, and GLK2 gene, and MYC gene. Okay, so we have some other method to verify if they are indeed involved in C4 chromosomal development. Okay, I won't have time to go through the detail for verification of these gene. But anyway, so we take, oh yeah, another important factor is, as I mentioned, some of the transcription factor is called ARF, auxin response factor. That means they have to have auxin produced to promote vascular development, cell division. So UCA is a gene, the key gene UCA on the top, on the top here. This gene promote auxin biosynthesis. Okay. So we want to put UCA gene, ARF gene, WAKI, and ARF3 into our transgenic rice. Okay. So we also do single cell transcriptome. This is really powerful approach because if you do leaf cross section of maize developing leaf, you can see the advancement of vascular. Initially, you can see 3 cells in the middle layer of maize leaf, and then develop into 5 cells, and then to even more 7 cells, and then this is another time point, and then this one eventually about 7 or 8 days later you have this. So we dissect these cells and then subject them to transcriptome, RNA messenger transcription, and then sequencing. And then we identify this model. In order for these stem cell for vascular development, they have to have a high auxin transport to this progenitor or stem cell for vascular development. So auxin has to come in, and this require LUX2 importer for auxin to come in. Okay, and then for the neighboring cell, they have to transport their auxin out of the cell. This pink one is exporter. Okay, so the neighboring cell donate their auxin to the progenitor cell of LUX for cancer development by this LUX2, this importer. Okay, so we also find that ARF control PIN1 expression, ARF4 control LUX2 and PIN1. So this is a very complicated situation. But anyway, the key point is that you need a higher auxin for this vent development. You need an importer, exporter of auxin. Okay, yeah. So after identification of these key transcription factor for Kranz anatomy, we have to use a 2 approach to verify their function. One is called loss function approach. We want to knock out these genes in C4 plant, like a maize, millet, and see what happened to Kranz anatomy. Okay, to be sure, indeed, they function in Kranz development. Okay, and of course, the second approach is gain of function approach. We want to take these genes and knock into C3 plant like a rice and see what happened to their leaf structure. Okay. Here I just quickly show you some of the result. Very interesting result. On top, this is a wild type. Usually you have 1, 2, 3, 4, 5, 6, 7 or 8 cells between 2 veins. Okay. And then if you overexpress SCR alone, not much, reduce mesophyll cell slightly from 8 to 6 to 7 cells. But if you overexpress in SHR, you see this drastic change in rice leaf. You can see they develop these very much like vascular cells all the way. It almost SHR promote bundle sheet development in this middle layer of rice leaf. So very striking difference. However, if you put these 2 genes together, they are back to normal. But if you add these two auxin transporter, the exporter, the importer, ping1, lux2, now we can reduce the mesophyll cell number by down to 5 to 6 cells. Okay, this is 2 transgenic line. So this is a small step to increase the vein density in rice. Okay. So I won't go through this, the same message from the last slide. Okay. So now we can reduce the mesophyll cell between 2 vascula in rice. Okay, from 8 cell down to 5 cells. Okay, but we still need to increase the chloroplast development in the bundle sheet cell of rice vascula. Okay, so that's our next goal. So as I mentioned earlier in my second talk, when we overexpress maize GLK2 or both GLK2 1 and 2 in transgenic rice, you can see increased, this is wild type cross section, and this is a transgenic. You can see increased chloroplast development, not only in mesophyll cell, but also in bundle sheet cell. So we hope this development in bundle sheet cell can be increased in the future. Okay, so this is our agenda for the future. But as I showed you this morning, with their own promoter, transgenic rice plant overexpression GLK1 and GLK2 can increase rice productivity up to 70%. Okay, so in the future we want to do this through cross hybridization. We want to stack the following gene into the same plant: functional C4 photosynthesis, high vein density, chloroplast development in bundle sheet cell, and for photosynthetic measurement and growth and granular analysis. Okay, so this is what we are doing. A final goal, a full C4 crunch anatomy in rice leaf. I think that this is a very long-term goal because we simply don't know very precisely how they regulate the rice vascular development. Okay, and the other important aspect is what regulate gene expression between mesophyll and bundle sheet cell in C4 plant. How we install that mechanism in rice. So these are the challenges ahead of us. Okay, so I wanted to acknowledge my collaborator. Early days in Japan, I worked with Makoto Nomura Agali, and then back in Taiwan, I worked with my major collaborator, Dr. Lee, and some other postdoc. And then in Jai University, I have probably more than 10 students involved in this project. Okay, so finally, you can see their happy faces here. Okay, so this is my presentation. I would be happy to entertain your questions. Okay.
Okay. Okay. Thank you, Prof. Maurice. It's very interesting research, and also it's very long time research, and I think it's still continue for the next maybe 5 or 10 years. Okay.
Yeah,
There is a question from a student. Her name is Ase. The first question is about, this is the first question is, in your opinion, is the C4 rice has a negative effect on the environment? And then the second question is about how long it took to get the C4 plants. And then the third question is, if we have a C4 plan, for the next time those plants can be changed to C3. And then the fourth question is,
maybe one by one. I couldn't take so many questions at one time. The
because
first
the
one.
question is in Indonesia. Is it a negative effect of C4 plant to the environment? Is there any negative effect in your opinion?
I don't think so. Actually, C4 plant would be very favorable, or the climate condition in Indonesia would favor C4 plant, because if you go to tropical, 95% of the plant species in this equator are C4 plant. Okay, and the number of C4 plant decreased towards a temperate zone. Okay, so I think as I mentioned, photorespiration increase at a high temperature. So in your country, like a tropical region, C3 plant is very inferior in photosynthesis. So if you can introduce a C4 mechanism to the plant, and they will adapt to higher temperature more effectively. So this is what I would say. Yeah. First question. Is that what he's asking, the student asking?
Yeah. And the second question is, how long it takes to make a C3 rice to C4 rice? How long it takes? It's maybe more than 30.
Yeah, I think that we have created what we call C3, C4 intermediate rice without Kranz anatomy. So as I mentioned, we still don't know very much about the regulation of Kranz anatomy right now. So when we put those genes, we don't see much change except reduce the mesophyll cell between two bundle sheet cells. So a little bit higher vein density in C3 rice, in the C3, C4 intermediate rice right now. So we want to combine these two traits, okay, higher vein density plus the C4 function in rice. And this may increase photosynthetic efficiency and water use efficiency, mineral use efficiency. This is a short-term goal. And for long-term goal, I don't know. I cannot predict if I'm going to see Kranz anatomy in C3 leaf. Really, I cannot answer. How long would take that to do? Maybe one day our technology advanced that we can have a better understanding, then it may be possible. Because in evolution, C4 plant evolved 60 times from C3 plant in both monocot and dicot, and in different phylogenetic branch with the same mechanism. So there must be some kind of master regulator allow them to appear in different evolutional branch. Okay, both monocot and dicot C4 plant evolve, and they evolve to adapt to tropical region. Okay, so we just simply don't know the mechanism at this point.
Okay, thank you. And then the third question, if for example, if we already have C4 rice and then we plant it in the field, is it possible that the C4 rice change again to the C3 rice?
Oh, okay. Very good question. In our experience, occasionally we do see the gene we introduce into rice will get lost. Occasionally, we don't know exactly the mechanism because we grow them side by side. Maybe hybridization occur, and then next generation in segregation, we just lost the gene of the maize gene in rice. But usually, I think that these genes are fairly stable in rice genome. We started up to maybe 6 or 7 generations after transformation, and they're still there. The genes still stably inherited. So I would say very low possibility of losing the gene and convert the rice back to C3, C4 or C4 rice back to C3 rice. Very, very low possibility.
Okay. And then the last question is, is it the C4 rice can dapat mengalami penurunan hasil? Can the C4 rice is will reduce the productivity?
What property?
And the C4 rice.
Oh, the grain
the C4
quality?
rice, is it possible that those C4 rice will reduce the productivity?
Productivity.
Yeah,
Oh,
the
no,
productivity
we
of
show
C4 rice.
Yeah, increase the productivity so far, but not every combination. Okay, I show you IP, both inorganic bicarbonate beneficial, PPC beneficial, and C carbonic anhydrase and K are beneficial. So right now we have IPCK transgenic plant, and we also try to induce G1, GLK1, GLK2 into the same rice plant right now. Yeah.
Okay.
So I think that the productivity will increase. Yeah.
Okay. It's not okay.
Yeah.
Okay, thank you Aseh for your question. Paling tidak berarti kalian ngerti lah dikit-dikit lah. Okay. Any other question? We still have 1 minute. Any other question? Okay. I think because of limited time, we have to stop the guest lecture. Thank you very much for Professor Maurice QPhD for sharing knowledge. It's very beneficial for us. I hope next time we will see maybe in research collaboration or in another place. Okay, thank you, Prof. Maurice. Saya kembalikan ke moderator, Mbak. Saya kembalikan ke MC, monggo Mbak.
Thank you to Professor Dr. Florentina Gusmiati as moderator and Professor Maurice Q, PhD as speaker. Ladies and gentlemen, before we close this guest lecture, we have a special performance from one of our faculty member. This is Java instrumental Tembang Pangkur. Will be performed by Dr. Budi Adi Kristanto. Please to Dr. Budi Adi Kristanto.
Bismillahirrahmanirrahim. Wah, ya. Baik. Sebentar. Bismillahirrahmanirrahim. Berubah mangsa hawa panas, udan sithik sawah tegale garing, dadi pusa kurang banyu ing wektu liya udan. Agawe banjir bandang hasil ngukup, dadi suda hasil pangan iklim ekstrim kang jalani, kinen mundak hasil beras, rine kaya genetike pari, ingkang C31 sini dikara berubah, tanduran dadi C4 ingkang unggul, tahan kering hasil kata, olah pikire Prof. Maurice. agawe parise 4 guna ngadepi perubahan iklim. Rob lan kering rusak iku ekosistem rusak. Teknik transkripsi fotosintesa jalur unduhane ilmu nyata dipaparke aneng Undip. Ya, matur nuwun.
Terima kasih Pak Budi. Monggo Mbak MC.
Thank you to Dr. Budiadi Kristanto. This is the ending of this lecture, Sustainable Agriculture in Facing Climate Change, Production of Seafood Rice, organized by Agroecotechnology Study Program, Department of Agriculture, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro. Thank you so much for your participation and see you on another occasion.
Ya, terima kasih. Thank you from Maurice. Terima kasih semua peserta. Semoga bermanfaat.
Terima kasih Prof. Kus. Prof. Maurisku sepertinya udah keluar.
Iya, kayaknya sudah keluar. Widi, apa kabarnya Widi? Ini mumpung ketemu Widi sama Agam ini. Oh, halo Widi.
Widi masih jalan ke labnya Prof. Ku katanya.
Oh, ya udah. Berarti sudah selesai ya. Terima kasih semuanya. Moga-moga bermanfaat. Paling tidak tahu ilmunya lah. Ya, oke.
Terima kasih Bu Kus, Pak Budi,
Ya, terima
juga Pak Ibu
kasih
semua yang
semuanya.
sudah hadir.
Ya, terima kasih.
Terima kasih.
Kita end meeting, J.